Hello, everyone, and welcome. You've joined us at the latest in public health research. This is our research seminar series at Berkeley Public Health. My name is Justin Rameis. I'm professor and chair of the Division of Environmental Health Sciences at the School of Public Health. Really delighted to see all of you. It's a great pleasure to welcome you today. I'm also joined by my colleagues, Professor Jennifer Ahern, Associate Dean for Research at the school, and Dr. Lauren Goldstein, who will help coordinate and facilitate Q&A after today's speaker. I'm also asked to remind students who are earning credit for the series to be sure to sign in. The link to do so is in the chat, so that you can be sure to indicate you're joining us today. In this research series, we hear from experts within our community in the School of Public Health at Berkeley who are leading critical research on topics that are most pressing for health in California and for the world. And today is really no exception to that. We're joined by Dr. Jennifer Head-Zudenegger. She is an infectious disease epidemiologist and also assistant researcher and PI of the Head-Zudenegger Lab in the Division of Environmental Health Sciences here at the School of Public Health. Her research group examines the roles of climate, public health policy, and individual behavior on infectious disease dynamics in a range of systems. She focuses on emerging infectious diseases like we'll hear about today, as well as zoonoses. Today, she's going to share her research on how climate and zoonotic hosts are contributing to the changing epidemiology of coccidioidomycosis, which is an infectious disease emerging in the southwestern US and in California in particular. Following her talk, we'll reserve time for your questions, so please hold on to those, and you can also share those in the chat. And Dr. Head-Zudenegger, welcome. You have the floor.
Awesome. Thank you so much for that introduction, Justin. And let me stop share because I think I just shared the wrong screen. But while I fix that, I am really excited to be here with all of you today and really excited to share the results of several years of research that we've been doing on valley fever in California. And as I'm pulling up these slides, if we were, in person, I would be asking all of you here today if you've ever heard of a disease called coccidioidomycosis. And I would expect that several of you have, but also that a good proportion of you have not. And myself, when I arrived to California in 2018, I had never heard of a disease called coccidioidomycosis. I have never heard of a disease called valley fever, but it is becoming a critically important fungal disease within the state. And various factors like climate change and rodents might be responsible for its changes in epidemiology. So what is coccidioidomycosis? Coccidioidomycosis, first and foremost, is the formal name for a disease we call valley fever, and we also sometimes call it cocci. And so you might hear me use those terms interchangeably throughout this presentation, but it is a fungal disease. It's caused by inhalation of the fungal spores from the Coccidioides species. These spores are endemic throughout much of the southwestern US, as well as parts of Mexico. In California, we have a species called Coccidioides immitis, whereas in the other states, it's predominantly Coccidioides possidacei. But in both species, the fungus lives and grows within the soil, and it derives its nutrients from material that's in the soil. And we'll talk a little bit later about what those nutrients might be. But as it grows in the soil, it can fragment and form individual spores. These spores become airborne when soil is disturbed or when wind creates fugitive dust emissions. These spores can then be inhaled by a mammalian host, such as a human, causing severe respiratory disease. For about 60% of people, there are no symptoms, but for the other 40%, it is a debilitating respiratory disease with symptoms that last a median of 42 days. And in about 5% of cases, the disease can become chronic. It's concerning because there's no vaccine against any fungal disease, and antifungal treatment is not very established in the state. So if we look at incidence rates across California, we see that the highest burden is within the Central Valley, this region that I've outlined here in white. Within that, we see the most in the lower parts of the San Joaquin Valley in counties like Kern and Kings. As we know, precipitation is very seasonal in California, with most of it falling in the winter. The Central Valley receives a pretty low amount of precipitation relatively in the winter. Temperature is also very seasonal, and this part of the state tends to be hotter during the summers than other parts of the state. While incidence is fluctuating year to year, on average, we've seen dramatic increases of incidence across California. So if we zone in here, as we're doing on patterns within Kern County, we see that incidence has increased over the past 2 decades. In this county, we've seen that the rates in 2018 are 8 times higher than the rates in 2000. What's even more surprising and concerning is that if we look at the pattern of incidence in other counties, we see even greater increases. So here I've pulled out rates from the northern San Joaquin Valley counties as well as the coastal counties. And in these areas, we're seeing that the incidence in 2018 was 15 times higher than the increase in 2000. This is a particularly surprising finding because these areas are not uniformly hot and arid. And that's kind of what we've thought of as the endemic territory for Coccidioides over the past several years. So what exactly is going on here? Amidst increasing incidence, we're also seeing increasing frequency and severity of drought in the Western United States. There was a recent paper that was published that actually describes that we are in fact in a mega drought that has been lasting since 2020, since 2000. And if we look at that 22-year period from 2000 to today, it's the driest period since 800 CE. Within this larger mega drought, we've seen smaller periods of very intense drought. So the 2007 to 2009 period was a period of very extreme drought for California, as was the 2012 to 2015 drought. So of course, with changes in disease and changes in climate comes a lot of speculation, a lot of which you'll see in the media. So these are just some examples of news reports that's speculating what's going to happen with valley fever and what is making valley fever surge. You know, this one's saying it's precipitation, this one's saying it's dust storms. And so there's a lot of speculation and a lot of it is based on research, but there's a lot that's unknown. And so let's consider what we do know and what we don't know about how climate affects Coccidioides. So if we look at the full life cycle of Coccidioides, we can imagine that temperature and precipitation likely affect each stage of this fungus's lifestyle differently. So first, if we look at these upstream factors, we know that upstream factors of temperature and precipitation might influence the amount of nutrients available in the soil, either via small mammals or vegetation, and it can also influence the soil microbial community that can act as competitors towards Coccidioides. Within this year prior, we know that precipitation can help the fungus grow. So most fungi require moisture to grow, and Coccidioides is likely similar to those other fungi. But then in this period right before dispersion, it's kind of a hot and dry conditions that cause those branching mycelia to break, forming spores that can become airborne. And so this sort of alternation between a wet period for growth and a dry period for dispersion is what's known as the grow and brillo hypothesis. And there's a good amount of support for it in the literature. We can also imagine that the joint influence of temperature and precipitation can hold unique influence. And this joint influence can often manifest itself as drought, because drought is a combination of both precipitation and temperature that leads to soil moisture deficits. And so while several studies have examined the influence of temperature and precipitation, the relationships that are reported in the literature vary quite wildly based on the location of the study, the specific lags before exposure that were examined, and also the model structures. What's more, there hasn't been any study that's looking at the causal effect of drought on incidence. And so the first half of this presentation will be focused on resolving these relationships. But before we dive into answering those questions, I want to throw in one more curveball into this life cycle diagram. And that is to consider what happens when the fungus is within the host. So here, let's take a mammalian host who has just inhaled a spore. That spore enters the lungs, where it can undergo replication and produce endospores within a larger structure called a spherule. If that spherule replicate or bursts, those endospores can then develop into new spherules and develop even more endospores. So in the human body, this process is very contained. But what if our host is something different? What if our host is a burrowing mammal that lives and dies within the soil? Could that replication of spores contribute back into the amplification of spores within the soil environment. And this question is what is forming the basis of what's called the small mammal endozoan hypothesis. This hypothesis operates under the premise that there are asymptomatic rodent hosts that contain spores within their lungs. The lung and the immune system, in order to contain these spores, develop granulomas around the spores. But when the rodent dies, which typically happens within a burrow, these granulomas dissolve, and so the spores can be released back. The spores, they're in the carcass of the rodent, and so it's hypothesized that this carcass serves as the primary source of nutrients for the fungus to grow and expand and branch throughout the soil. This hypothesis has actually been long in the making, but has a somewhat long and tumultuous history. It was first introduced way back in the 1930s when a man named Chester Emmons confirmed that rodents were infected with Coccidioides at relatively high prevalences. And at that time, he suggested that rodents are actually zoonotic hosts for Coccidioides rather than just incidental hosts. This observation was then supported by studies that demonstrated that Coccidioides was detected in higher concentrations in rodent burrows compared to surface soils. However, a study that happened in the late 1950s, where there was an outbreak of Coccidioides, somewhat dissuaded people from believing in this hypothesis. And so what happened was there was an outbreak of Coccidioides that was near a midden, which is where rodents might pile their refuse. And so researchers went and they tested the rodents for Coccidioides, and they found that none of the rodents were infected, but that many of the soil samples were positive. And so that was the study that really squashed the rodent hypothesis and led researchers to conclude that soil is what is the primary environment for Coccidioides. And then over the next several years, most of the studies that examined Coccidioides in the environment did so investigating different characteristics of the soil that predicted its presence. So factors like salinity were considered. At this time, there were a lot of other studies that did show strong associations between burrows and Coccidioides, but of course, burrows themselves are unique soil environments. In the 2000s, there was a groundbreaking study that looked at the genomes of Coccidioides and actually found that they had evolved to contain genes that degrade animal keratin. And they had actually lost genes that degraded plant matter. And so this sort of began to shift the hypothesis back to suggest that maybe Coccidioides is not degrading decayed plant in the environment like many fungi do, but is actually degrading the animal protein itself. And this led to the publishment of the endozoan reservoir hypothesis in 2019. And so throughout this presentation, I will attempt to address kind of three main gaps in our knowledge of the epidemiology of coccidioidomycosis through these three various studies that I'll talk about. The first is to determine how prior sequences of temperature and precipitation affect incidence, and to examine how these effects differ based on regions in California. The second study is to determine the causal effect of the two major droughts in the past 2 decades on incidence in California, and again, compare how this effect varies across regions. And then the last is to identify the association between rodents and Coccidioides, considering how burrows and soil conditions might mediate the relationship. So I'll jump into the first study, which is looking at how sequences of temperature and precipitation affect incidence. For this analysis, we used a distributed lag nonlinear modeling approach. We started by linking information on monthly cases per census tract to information on environmental variables, including lag, temperature, and precipitation, where temperature and precipitation were lagged by up to 36 months. Because Coccidioides is quite seasonal in California, peaking in the fall months, we restricted this analysis to September through November, which also allowed us to map the lagged predictors of interest onto seasons of the years. So let me orient you to this diagram here, which is what we'll be using as a template to show our results. On the right-hand side is the disease onset, and it's restricted to the fall months. The x-axis tracks the environmental conditions back in time from onset. So the 1 here corresponds to 1 month prior to onset, and the month 36 corresponds to 3 years prior to onset. We then regressed monthly census tracked cases against the lagged precipitation and temperatures using splines to account for nonlinear exposure response relationships. We controlled for other environmental variables like elevation and soil texture. And we ran one of these models for each county. And then we pooled the exposure response relationships using a meta-analysis. So what this new plot is showing here is a pooled exposure response relationship for the effect of precipitation on incidence, where precipitation is lagged by 3 months. So it reflects summer precipitation. We then examined where the 25th percentile and the 75th percentile of the variable of interest was, and computed the incidence rate ratio associated with increasing that variable from the 25th to the 75th percentile. So if you look here at this plot, the 25th percentile for precipitation is 0 for summer, and the 75th is about 2.5, and this is associated with an incidence rate ratio of 0.95. And that is what we're mapping here or plotting here on the other diagram. So when is precipitation associated with incidence? We found that in the 1 to 4 months prior to disease onset, more precipitation was associated with fewer cases. And if we consider the life cycle of Coccidioides, which is reflected here in the upper right-hand corner, we think that this 1 to 4 months prior to onset is likely corresponding to when the mycelia are breaking into individual arthrochonidia and releasing and dispersing throughout the environment. If we look at the 5 to 10 months prior to onset, which are the months that correspond with spring and winter, we see that higher rain is associated with more incidence. We expect that this is probably the part of the life cycle that is associated with the mycelial growth, the part that requires moisture. If we look back past 1 year prior to onset, we see that the effect of precipitation is more variable, but generally higher rainfall is associated with fewer cases. What is happening here probably corresponds to influences on these upstream factors influencing growth. However, what was more interesting here was that the conditions that happened in these 1 to 3 years prior to onset modified the effect of these more recent environmental variables that happened in the 12 months prior to exposure. So we found, again, recalling that the wet winter increases incidence, we found that when that wet winter followed a dry winter, the effect of that wet winter was enhanced by 19%. And if a wet winter followed 2 consecutive dry winters, the effect of that wet winter was enhanced by 36%. I won't go through the results from temperature in as much detail just due to time, but I also want to highlight that we found that hot summers and falls, so this period of time right before exposure, was associated with higher incidence, but that cooler winters and springs were associated with more incidence. We then examined how each of the county level exposure response relationships varied based on the county's typical profiles of their precipitation and temperature. So these graphs here are showing an exposure response relationship between winter rainfall on the left and summer temperature on the right, and coccidioidomycosis incidence. Each of these lines is representing a different county, where the counties are colored by their median level of rainfall or temperature. So if we restrict to just looking at the hot and dry counties, we find that these counties, incidence is strongly influenced by rainfall, but it's not as much influenced by temperature. And these counties tend to be the counties that have had the highest incidence rates of cocci, so counties like Kern and Kings. But if we restrict to looking at the cooler and wetter counties like those in the Northern Valley and along the coast, we see that they're not so much, incidence is not so much affected by precipitation, but it's very much influenced by heat. And so what this suggests to us is that different counties have different rate limiting steps in the life cycle of Coccidioides. The dry counties often experience conditions hot enough to help the fungus disperse, but they might not experience enough rainfall to help the fungus grow. Whereas cooler, wetter counties likely experience enough rainfall for growth, but they might not have enough heat for that fragmentation and dispersion process. So to conclude from this part of the analysis, we find that in the short term, hot and dry conditions promote incidence, and the cooler counties are especially sensitive to heat at this time. A little bit more upstream, wet conditions promote incidence, so wet winters. And we found that drier counties are especially sensitive to precipitation during the winter. We also see that consecutive dry years enhances the effect of a wet winter, likely through influence on the upstream factors. And so this brings us to ask, what about drought, since drought produces consecutive periods of dry years? And so that leads us to the second part of our analysis, which is determining the causal effect of the 2007 to 2009 drought and the 2012 to 2015 drought. So for this analysis, we modeled year-round incidence, and we did so using an ensemble prediction algorithm. So first, we generated various algorithms ranging from very simple generalized linear models to more complex random forest models. We calculated the leave out 1 year cross-validation error, and then we weighted the different candidate algorithms by their out-of-sample prediction error in order to compute an ensemble model that minimized out-of-sample prediction error. So this is just one example of a predicted model from the western part of Kern County. This graph is showing the actual observed number of cases per month as dots, and then the line represents our fitted model. And so you can see here that our model prediction did quite well. And for all the counties that we included in our analysis, the R-squared of the model was above 0.9. We then used a G-computation approach to estimate incidence under a counterfactual no drought scenario. And so we used our fitted ensemble model to calculate the expected number of cases under the observed conditions of rainfall and temperature, and then to calculate the expected number of cases under a counterfactual scenario, where any rainfall that was observed below average during the drought period was set to the average, and any temperatures observed above average during the drought period was set to the average. We then subtracted these two estimates of cases to come up with our target parameter, which was the difference in the number of cases that could be attributable to the drought. So again, this is showing the example from Western Kern County. So the dots are still the observed data. The black line is still the fitted model. And the colored lines represent the incidence that we would have estimated in absence of drought. And so if you look here at the blue line, we see that during the 2007 to 2009 drought, we expect that we would have seen more cases during drought if that drought had not occurred. But that following the drought, we would have expected to see a little bit less cases. So we completed this process for all the counties in our study region, and we summed across all the counties to come up with the total effect. And what we found was that we experienced fewer than expected cases during the drought than we would have expected if the drought had not occurred. And this was true of both droughts. However, in the 2 years that followed the drought, we actually saw an excess of cases, more than we would have seen if the drought had not occurred. And for both of these droughts, the excess in cases that was observed more than offset the amount that declined during the drought. We then examined how this effect varied across counties. So Kern, which is shown by the arrows here, is hot and arid, and it experienced large declines during the drought, the largest relative declines out of any other area that was seen. However, if we look at what happened after the drought, Kern experienced relatively mild increases, whereas counties along the coast and in the northern San Joaquin Valley experienced much larger percent increases than expected. And so if we examine this in terms of absolute cases, we calculate that Kern actually had cases that were averted due to drought overall, but that the counties along the coast and in the northern San Joaquin Valley experienced the greatest net increases. So to summarize the findings of this analysis, we found that drought displaces and then amplifies incidence, with the greatest amplification occurring in these northern San Joaquin counties and the coastal counties. And if we recall from the beginning of the presentation, these are the counties that have experienced the biggest percent increases in incidence of coccidioidomycosis over the past 2 decades. We know from climate modeling that the frequency and severity of drought is expected to increase in the Western United States. And so what this suggests to us is that this expansion of Coccidioides into these wetter and cooler areas might continue in the decades to come. The good news, if there is good news, is that some of these areas that are already very hot and dry might actually see a decline in cases due to this increasing aridity. And so I'd like to discuss some strengths and limitations of both of these climate analyses. This is the first study to apply causal inference techniques to answer the question of drought and incidence. We do so by linking incidence from census tracts to environmental exposures. And this is actually the first study to do that as well. All the previous studies have examined county level incidence and environmental exposures. And then we use very flexible model structures, which enables us to capture these complex interactions and nonlinear relationships between climate and incidence. There are some limitations. Cases are assigned to the month and estimated date of disease onset, but actually there is a pretty long lag in time between when a case might experience symptoms and when they report to the healthcare system. And so the lags might capture very different timescales there. Cases are assigned to the census tract of residents, and so we don't actually know if they were exposed in their census tract of residence or somewhere else, especially because this disease is very occupationally associated. And then the last limitation is that drought can be defined in multiple manners. There's very different types of drought. The one we're considering here is that which is defined by soil moisture, but there's other ways to define drought, and so future study might consider how different drought definitions might affect these sorts of estimates. So now I'd like to go on to kind of a slightly different part of this study, which is examining the association between rodents and coccidioides. And so it's been a while since we talked about the small mammal endozoan hypothesis. So I just want to remind listeners a little bit of what it is again. So recall this hypothesis is that rodents inhale spores, they wall off the spores in granulomas inside their lungs, and when they die, the granulomas dissolve, releasing spores. The spores use the rodent carcass as a source of nutrients to grow and propagate throughout the soil. And beyond kind of genomic analyses that have suggested that Coccidioides actually prefers to degrade animal keratin rather than plant matter, one of the key pieces of evidence supporting this hypothesis is the strong association published between burrows and detection of Coccidioides in the environment. This connection between burrows and Coccidioides is reflected in several studies, but it's hard to know just how much the influence is because of rodent burrows or because of the influence that burrows has on modifying soil conditions. So burrows have very different soil conditions than other surface soils. They kind of create a nice homeostasis, and this could influence Coccidioides' presence. The challenge here is that we really want to know about the effects of rodents, not burrows. And so we want to ask, is it the rodents, the soil conditions, or the burrows that is most responsible for determining Coccidioides' presence in the environment? Unfortunately, it's really hard to disentangle a rodent from a burrow. And in the past, we have been challenged in our ability to do so. And so no study has been able to do so yet. And this is where the Carrizo Plain study enters. So the Carrizo Plain is the largest intact grassland in California. It's located just west of Bakersfield and Taft, and it is unfortunately a site of many outbreaks of valley fever, including among researchers from UC Berkeley who go to the Carrizo Plain to conduct research. If you've heard of the Carrizo Plain, it's probably because it gets really amazing super blooms. And if you look up Carrizo Plain on Instagram, you'll see just these incredible photos of wildflowers that are just covering the hills like a painting, and it's really amazing. But as we know, Instagram is not always reality, and California is in a drought. And so all the times that we have visited the Carrizo Plain, it in fact looked like this, which is, still beautiful in its own way, but very different. The Carrizo Plain was established as a national monument due to its large population of multiple endangered species, including the kit fox, which is shown here on the right, upper right. Many of the animals that are there are either those that burrow or those that fly. And the most important mammal for the purposes of our study is this mammal in the upper left-hand corner, which is called the kangaroo rat. These kangaroo rats are known as ecosystem engineers because they build the burrows that all the other animals use, including the larger burrowing mammals like the kit fox and the badgers. The Carrizo Plain is also home to many research projects, and one of these research projects is the Carrizo Plain Ecology Project. This project was established in 2014 in order to study the effects of the kangaroo rat on vegetation. And so at that time, they created these experimental plots where the center of each experimental plot contained a rodent exclosure. And so these fences were built, these 20 by 20 meter fences, were installed in order to exclude rodents from entering this 20 by 20 plot of land. So here is showing the location of the Carrizo Plain in California. The middle picture is showing one of these experimental plots from space. So you can see where the larger plot is located, and then the little exclosure is shown in the middle. And then if you look at this picture from space, you can see that the vegetation is very patchy throughout the environment. And that is because this landscape is actually punctuated by these rodent precincts. So the giant kangaroo rat is a precinct forming rodent, which means it builds its burrows in these sort of interconnected systems that form a sort of mound. And they clip the vegetation in and around these burrow entrances, which results in less vegetation on these precincts. And so because these rodent exclosures were established, we're permitted for the first time to disentangle the effects of rodents from the effects of burrows, because inside those exclosures, we have burrows that do not contain any rodents, and in theory have not contained any rodents since 2007. And so we have been sampling soil seasonally across a factorial design that crosses burrows and surface soils with rodent exclosures and non-exclosures. In each season, we've collected 500 samples, and we've collected across all 4 seasons and April twice. So totally we have 2,500 samples collected. Along with collecting the soil sample, we collect information on the vegetation around the sample, soil moisture, and soil temperature. Another research team, which is shown here, does rodent abundance surveys where they do capture, recapture on the rodents and estimate the number of rodents per plot. So this represents an example plot, and I'll talk through now how we've selected each of our samples. So as you can see, the plot is 140 meters by 140 meters with the exclosure in the middle, and it's dotted by these precincts, which either look active or a little bit less active. So when we first arrived at the plot, we divided it into a grid, and we randomly selected 2 grids by a random number generator. We went to the location of each of those grids, and outside the exclosure, we identified 2 of the closest precincts that looked active. And inside the exclosure, we identified a precinct that looked inactive, since it should look inactive. Then we collected 5 samples from within the burrows of each of the selected precincts. And we also collected 5 surface samples marching outward from those precincts. And so in total, we have 20 plots. We have 5 clusters per plot and 5 samples per cluster. And this is just another diagram showing where we've collected geographically. So then we brought our samples back to the lab, we transferred the soils, extracted the DNA, and ran qPCR on them in order to detect Coccidioides. We then did a mediation analysis with G computation following the approach that was published by Wang and Arya in order to determine the total effect of rodents and the indirect effect of rodents on Coccidioides. So just to orient everybody to some terms that I'll use in the slides to come, we're considering the rodents as our main exposure of interest, which is indicated by an X, the burrows as the mediators of interest, M, and Coccidioides in soil as a binary indicator as our outcome Y. In another round of analyses, we also include soil conditions, which is represented by Z. So our target parameters of interest is the total indirect and direct effect of rodents on Coccidioides, as well as the percent of the total effect of rodents that's acting via burrows. So I encourage everyone to read the paper by Wang and Arya if you're interested in finding out more about the exact method of how to do this mediation analysis. But briefly, we created two models. The first was to model the mediator conditional on the exposure and the covariate, so the expected value of burrow conditional on rodents. So in our experimental design, we were able to have burrows where rodents did not exist, but in real life, this doesn't actually happen. And so we deterministically set values for this equation. So if X was 0, meaning rodents was 0, we set the value of burrow equal to 0. But if rodents were present, meaning X is 1, we set the value of burrow, the probability of a burrow to 0.5, meaning there could be either a burrow or a surface soil. We next modeled the expected value of the outcome conditional on the burrow, the mediator, the burrow, which is the mediator, the rodents, the exposure, and then other covariates. And because our data was clustered, we did this using a generalized linear mixed model. We then used our models to simulate values for the mediator and the outcome using drawn values of X's, and then we fit marginal structural models, which gave us our target parameters of interest. So I'll go through some of our results. To date, we have analyzed 1,041 samples in the lab, and so these results are still preliminary, but we found that almost 18% of our samples are positive for Coccidioides. And if we look at the prevalence across sample type, we see that by far the greatest prevalence of Coccidioides is found in burrows that contain rodents. Over 30% of those samples were positive for Coccidioides. About 20% of the samples taken from burrows within the exclosure were positive for Coccidioides. We did not find any positive samples in surface soils taken from the exclosures where rodents don't exist. And only about 3% of our samples from surface soils in areas where rodents were positive for Coccidioides. We do find significant differences in soil characteristic based on burrows versus surface soils. In general, we found that burrows have lower soil moisture than other surface soils, which was what we expected based on the literature. We also took continuous measurements of temperature within the burrows and the surface soils, and we found that burrows really help buffer temperature extremes. So during the heat of the day, the temperature within a burrow is much less than the temperature in the surface soil. And moreover, the temperature in a burrow with a rodent is even more buffered during the heat of the day. This table is showing the result of our generalized linear mixed models, predicting the expected value of Coccidioides presence given burrows and rodents. And we find here that even controlling for soil conditions and sample type, the odds of Coccidioides presence where rodents are present is 4.3 times higher than the odds of detecting Coccidioides where rodents are absent. We do see also very large odds ratios associated with burrows as well. So controlling for soil conditions and rodents, we see that the odds of detecting Coccidioides is 22 times higher in a burrow compared to in surface soils. We also see that higher soil moisture is associated with lower probability of detecting Coccidioides. So this slide shows some results from our first mediation analyses where we just examined rodents and burrows. And we find our total effect is 58. With wide confidence intervals. And so this means that the odds of coccidioides is 58 times higher where rodents are present compared to where rodents are absent. And we estimate that about 66% of this effect of rodents on coccidioides presence is mediated via burrows, meaning that about 1/3 of the total effect is direct and not mediated via burrows. So we repeated this analysis controlling for soil conditions. And how these results can be interpreted is after you suck up that variation of the burrows contributing to changes in soil conditions, or also the rodents contributing to changes in soil conditions. And we see here fairly similar results, but the amount of the effect of rodents that's mediated from burrows does decrease a little bit, meaning that the direct effect of rodents increases proportionally. We also conducted some mediation analyses to examine the effect of rodents and burrows on soil conditions. And we found that soil moisture is predominantly mediated via burrows, whereas vegetation levels are predominantly controlled by rodents. And that temperature is both affected by rodents and burrows. So to put some of these findings in a summary, we find that we actually don't detect coccidioides very much if we don't have a burrow and a rodent. So the odds of detecting coccidioides when rodents are present, we found to be 58 times higher than when rodents are absent. This is under the assumption of our mediation analysis that you need a rodent to make a burrow. We also found that 33% of the association between rodents and coccidioides is direct, which suggests that rodents themselves do have important advantages that it's conveying to coccidioides above and beyond just making a nice environment through a burrow. However, there is still a very strong indirect effect of burrows, which does suggest that burrows are important microhabitats that do support soil conditions conducive to coccidioides. And some of these soil conditions might be the lower soil moisture that's found in burrows. So this has been the first study to start to disentangle the rodent from the burrow, and also uses a highly systematic experimental design to start to ask these questions and enable the performing of a mediation analysis. However, there are still some limitations of this analysis and additional studies that we'd like to explore. We didn't measure other abiotic factors that might hold influence, like humidity. There could be other factors at play here, like nutrient profiles within burrows that have rodents. So one question we'd like to ask is, is it just the presence of more keratin in burrows, or is it actually the presence of the decayed rodent carcasses in the burrows? We were not able to confirm whether burrows were truly active versus inactive. And we know from prior study that spores can remain viable for 6 years or more. So even in these rodent exclosures, there could be coccidioides that's just kind of hanging out as a spore, but not actually growing in any meaningful way. So just to kind of try to tie our findings on climate from the earlier half of this presentation to our new findings on rodents, we think that there's a lot of room for, speculation here and a lot more research that could be done. We found that wet winters are supporting coccidioidomycosis incidence in the fall. And when we talked about it, in the first part of our presentation, we talked about it kind of as a direct relationship, but we also know that what conditions support vegetation growth, which support rodent populations, but that during drought, rodent die-off is highest, which might mean that during drought is when you're having rodent carcasses contributing spores back into the soil. So these are all directions that I think remain to be investigated. So I'd like to conclude by just describing how we've updated our understanding of the role of climate and rodents on the shifting epidemiology of valley fever. So we've seen that valley fever is an emerging fungal disease in the American Southwest, and we now know that drought might be contributing to this expansion by leading to increased case counts in the years following drought. We've seen that incidence is increasing most rapidly in wetter and cooler counties, and that these counties are really sensitive to variation in temperature and heat. And so our results are suggesting that as California moves to a hotter and more arid region, these wetter and cooler counties are really affected. The good news, as I mentioned before, is that increasing aridity might actually lead to declines in incidence in some of these already very dry counties like Kern. Finally, the reservoir for Coccidioides has been under debate for many years now. Is it the soil or is it a rodent? Our results suggest that burrows do create suitable microhabitats for Coccidioides, but that the presence of rodents greatly enhances detection of Coccidioides. So I'd like to just conclude by thanking an incredible team of researchers here at Berkeley for their support, California Department of Public Health for their partnership in obtaining the incidence data and working with us through understanding this disease, our climate modelers at UCLA and Texas Tech, and then the Carrizo Plain team at California Poly. And yeah, I'm happy to take any questions.
Great, thank you so much, Jennifer. That was really, really fascinating and really appreciate your talk. Really interesting work. Thank you for sharing it. So if people want to add questions in the chat, feel free to put them in the chat. Here's one question. Can you touch briefly on the public health implications or clinical significance of your findings?
Yes, absolutely. So valley fever is a bit of a tricky disease because there's, as I mentioned, there's no vaccine for it. And treatment can be done with antifungals, but it's not always effective. And so really the best tool we have right now for preventing and treating valley fever is awareness, both awareness of, when and where incidence might be highest, and then also awareness of, if I'm feeling sick, could it be valley fever? And so I think that the findings, especially on climate, can help both individuals living in the regions understand, is this a big year for valley fever? And then can be, more prepared to go seek care if they're experiencing respiratory symptoms. And the physicians can also be aware and think about diagnosing valley fever. Because even though this disease, is increasing in the state, the physician awareness is actually quite low, as well as the awareness in the general public. So for instance, I think coming out of this drought, we're going to see a really big year of valley fever as soon as the rains return. And so people should be really aware that if they're experiencing cough, they should seek a healthcare provider immediately and ask about, testing for valley fever and then getting them on prompt treatment. I think the findings on rodents can also have important implications, especially for occupational workers in California. Valley fever is an occupationally associated diseases, and so certain occupations do have to teach their employees about the risks of valley fever and provide the option of masks. And so a lot of construction workers are working around rodent burrows, or due to great environmental protection rules in California, if there's a construction project and there's an endangered rodent species, they have to relocate those rodent species. And so knowing that if you're a worker who's digging in an area where there's rodent burrows, you should take extra precautions and perhaps wear a mask if you can, or just be aware of the risk you have for getting sick.
Great, thank you. I see that Jacob, you have your hand raised. Do you want to ask your question?
Oh, sure. Thanks so much for the presentation. I was, so you get into this a little bit in your answer to the last question, but I was wondering if you would talk more about how you might be thinking about exposure behaviors related to this disease. It seems like that could also mediate the effect of these sort of, you have the ecological risk, but then there's how are the people actually interfacing with the ecological risk, and has that changed in any way over time? I know you mentioned the occupational health risks, but I'm curious to hear more about what you're thinking about that.
Yeah, that's a really great question. I think it's something that we're thinking about, but we're not quite sure how to address yet in research. And so we would love to have other ideas, especially from, people who are more versed in social epi and demography. awareness is increasing, and with the recognition of this as an occupationally associated disease, there are protections that are afforded to individuals. Unfortunately, the Central Valley is a hot area, and if you're doing a lot of hard work, you don't really want to be wearing an N95 mask, and so changing exposures that way is a bit tricky. I guess we've been thinking to as well about how climate influences behaviors, and one example is it will influence, we expect, agricultural patterns in the state. Right now, California is undergoing a large fading out of agricultural land due to drought in order to conserve groundwater resources, and so we do expect that these sorts of like drought will affect behaviors, will affect exposure, but we haven't robustly modeled that yet, even though we'd like to, and we think it's important.
Great, thank you. I know it's just about time, but there's 2 other questions if people can stay on, and if you have to hop off, I understand. Divya wanted to know, if someone lives in an area prone to valley fever, what are some precautions they can take with to avoid getting sick? She had a relative get sick with valley fever in Arizona, and they'd like to know how to prevent recurrence.
Yeah, it's a great question, and it's difficult because we expect that you can get sick just by breathing in a spore in the air. And so if you are doing an activity like digging in the soil, especially in an endemic area and around burrows, we would recommend taking appropriate precautions such as wearing a mask. At this point, I think the best recommendation we can have is just increasing awareness, because there are antifungals that do work better if you're able to catch the disease faster. And then there is a vaccine that's under development right now. It's being tested on dogs and seems to be having good effect. And so hopefully in the years to come, we can add, get the vaccine to this list of precautions.
Great. Well, thank you so much, Jennifer. We really appreciate you presenting today. And thanks everybody for joining. And hope everyone has a great day. Thank you.
Thank you.
Bye-bye.